Welcome back, biologists. In this screencast, we're going to begin Chapter 9 and talk about cell division and mitosis. In this chapter's case study, we're going to talk about cancer and how we can use a drug called Taxol to fight it. Taxol is a derivative from the Pacific yew tree. This chapter is about cell division, and cancer is a disease where cells divide uncontrollably. The body is no longer able to regulate cell division in these cells. Cancer cells divide inappropriately and can accumulate. In these cases, they can form tumors. The photo on this slide shows cancer cells in the middle, surrounded by normal cells. Cancer cells are physically and functionally abnormal. This pie chart shows how cancer is the number 2 cause of death in the United States, in between heart disease, the number 1 cause of death, and strokes, the number 3 cause of death. After cancer has been diagnosed, there's usually 3 different ways in which the cancer can be treated. First is surgery, where the cancer is physically removed. Then there's radiation, where focused energy is used to kill cancer cells. And then there's chemotherapy, where chemicals are used to kill cancer cells. Taxol is an example of a chemotherapy drug, a chemical used to kill cancer cells or to slow down their growth and division. Taxol has been shown to be highly effective against certain types of cancer and is found in the bark of the Pacific yew tree. The chemical structure of Taxol is shown on the left. A Pacific yew tree is shown on the right. This diagram shows the geographical range of the Pacific yew tree. It can be found in the Pacific Northwest of the United States and in Canada. Taxol, like most chemotherapy drugs, works by interfering with cell division. Cell division is the process by which cells reproduce themselves. Cell division is important for our normal growth and development. It's also important for the replacement and repair of damaged and aged cells and tissues. Many of our cells don't live forever, and we depend on cell division to replace them. For example, stomach cells are replaced every 2 to 9 days. Blood cells are replaced every 4 months or so. As I mentioned before, cell division is important for our growth and development. We begin life as I mentioned before, cell division is important for our growth and development. We begin life as one fertilized cell. Through cell division, that one cell becomes the trillions that make up our body. Cell division is important for cell replacement, for cells in our body that don't last our entire lifetimes. The infographic shows how cells in our bone marrow divide to create new blood cells to replace old blood cells that have reached the end of their lifespans. Cell division also allows our wounds to heal. The infographic shows that when we have an open wound, cells will divide until they come back into contact with other cells. Let's take a look at how cells divide. In cell division, cells do not simply split in half. They go through a series of phases called the cell cycle. The cell cycle is ordered and predictable. It has a preparatory part where the cell prepares to divide, and a division part where the cell divides. Through the cell cycle, one cell divides to form 2 identical cells. In the preparatory phase of cell division, cells will copy their contents. They'll duplicate their organelles and DNA. They'll also produce additional cytoplasm. The purpose of the preparatory phase is to make sure that each new cell has the same amount of these things as the original cell. The preparatory phase of cell division is also known as interphase. Interphase can be broken down additionally into the G1, S, and G2 phases. In the G1 phase, the cell will grow in size. it will also produce additional organelles and cytoplasm. In the S phase, cells will copy their DNA. At the end of the S phase, each chromosome has a duplicate copy of itself. The chromosome and its duplicate stay connected at a location called the centromere. They're also called sister chromatids at this point. During the G2 phase, the cell further prepares for division. After the preparatory phase, which we also call interphase, cells will enter the division phase. The division phase can be broken down into mitosis and cytokinesis. The diagram on this slide shows how interphase can be broken down into the G1, S, and G2 phases, and the division phase can be broken down into mitosis and cytokinesis. It also shows on the right side how one chromosome can copy itself to form two sister chromatids that stay connected at the centromere. During mitosis, these two sister chromatids will separate and make their way into two different cells. During mitosis, chromosomes will evenly divide themselves amongst two new cells. A chromosome and its copy are called sister chromatids. They're connected and will separate during mitosis at the centromere. This is how a cell with 46 chromosomes can divide and form two new cells with 46 chromosomes each. Cytokinesis is the final stage of the division phase of the cell cycle. In cytokinesis, the enlarged cell will split to form two new cells. Each new cell will have a full complement of DNA, or a full set of chromosomes. The cell that we start with at the beginning of the cell cycle is called the parent cell. At the end of the cell cycle, when we have two cells, those cells are called daughter cells. Let's take a closer look at mitosis. Mitosis itself can be further divided into a series of phases or steps. Mitosis and cytokinesis make up the division phase. The division phase can also be referred to more simply as the M phase. You'll notice a series of boxes in your packet. As we review each phase, try and sketch it in your notes. Before mitosis and the M phase begin, we have interphase. During interphase, chromosomes will copy themselves to form sister chromatids. The sister chromatids are found in the nucleus. The first stage of mitosis is called prophase. During prophase, the chromosomes will condense. The nuclear envelope or membrane will start to break down. Microtubules in the cell will form a mitotic spindle. The microtubules and the mitotic spindle will help the chromosome separate later. The second stage of mitosis is called metaphase. During metaphase, microtubules will attach to the sister chromatids. The sister chromatids will line up in the middle of the cell. The chromosomes are setting themselves up to be evenly divided into two new cells. Anaphase is the third stage of mitosis. In anaphase, the microtubules that have attached to the sister chromatids shorten, pulling the sister chromatids to opposite ends of the cell. The fourth and final stage of mitosis is telophase. In telophase, the sister chromatids have separated and can be found on opposite sides of the cell. The mitotic spindle disassembles. New nuclear membranes or envelopes form around each set of chromosomes. These become the two new nuclei of the two new cells. Cytokinesis occurs after mitosis. In cytokinesis, the cell membrane pinches and splits to form two new daughter cells. In plants, a new cell wall also forms between the two new daughter cells.